Hi everyone. I hope you're doing well. Thanks for joining us. This is, as Karen said, I think the sixth talk in our Vicky Funk seminar series. And we're really happy in the Biogeography Society to be able to provide this. So the series is named in honor of Vicky Funk, who was a systematist and biogeographer specializing in the sunflower family. She was a highly influential scientist within her fields of study, advancing many aspects of plant biogeography and her particular group. But she also was super influential in thinking about sort of general biogeography patterns. She had a very influential co-edited book with Wagner on patterns of evolution and biogeography in Hawaii. She was also noteworthy for her professional service, which she did a lot of. And she served on a lot of academic societies and led a lot of academic societies. She was a founding member of the Biogeography Society. She was part of a small group of scientists that met in Santa Barbara, California in 2000 and 2001 with the aim of establishing the International Biogeography Society. A few years after the society was founded, she became one of its first presidents. What I remember about her most was her infectious energy. She just was sort of boundless in what she could do, and she had this can-do attitude and enthusiasm that I think was really important early on when the society faced a lot of barriers to getting founded. I think that her energy, intellect, and wisdom are a big part of the reason that IBS exists today. And I'm glad that we have this series named in her honor. So in that context, it's a special privilege to be able to welcome Michael Sundue to give today's talk, as he's a plant systematist and biogeographer like Vicky. Michael is assistant curator at the University of Vermont's Pringle Herbarium. He did his PhD at the New York Botanical Garden in collaboration with the City University of New York. And he studies the evolution, systematics, and biogeography of ferns, focusing particularly on tropical ferns. He is an interesting scientist in sort of the sort of spatial scales and questions that he spans. Everything from latitudinal gradients in diversity to patterns of montane diversity in the tropics and global patterns, all the way down to looking at particular genera and families. And he's also spent a lot of time doing field work in places like New Guinea. He's had some pretty influential research, but besides his research, he's also having a big impact by helping to lead the website Ferns of the World. And I'm delighted to have him here today. So Michael, I'm going to turn it over to you.
Great, thanks, Dov. Thanks everyone for having me. Before I share my screen, I'll just say, yeah, it's a privilege to be here. I'm delighted. It's an honor to speak in the Vicky Funk lecture series. I never worked directly with Vicky, but she was such a generous and enthusiastic colleague. She put me up at her house the first time I went to the Smithsonian. She took me out to lunch when I was a graduate student. Things like that, I will never forget. So I appreciate that. And with that, I will share my screen. So yeah, and I'm also, I'm thrilled to present this work, and I'm so thankful that the Society invited me to present this particular paper, because as Dov says, I work at a lot of different scales. Often, I spend the day trying to identify a specimen, or spend the week or the month in the field collecting material. Other times we're synthesizing that work, and this project was really fun because it was a project where we got to synthesize from the ground up many thousands of records, representing the work of thousands of people into a big picture so that we can get, the global view of fern diversity. And, the intellectual inspiration here is really based on this simple idea that a lot of people have been talking about recently, which is the uneven distribution of life on Earth, right? This has fascinated people for centuries for good reason. Why are the tropics so much more diverse than temperate regions? Two of my inspirations, and I'm sure those of many people in this group, were Humboldt and Bonpland, who during their American expedition were among the first to try to synthesize latitudinal and elevational diversity gradients and explain the relationship between climate, geography, and vegetation. Their expedition traveled to Mount Chimborazo in Ecuador. Maybe you've been there. It's a well-known and famous large volcano almost directly on the equator. So Humboldt published his iconic depiction of Mount Chimborazo, seen here on the left, in his essay on the geography of plants. And you can see how they handled this. They did an expedition ascending the volcano. They illustrated the vegetation zones they saw along the way. And in that white space in the illustration, you can see all the names, the scientific names of plants that they saw along the way. They wrote down at the correct elevation where they saw different species, botanizing the whole way up. And what they remarked on was how certain elevations reminded them of other places they had been on Earth. Like, oh, this reminds us of the Swiss Alps. So they were fascinated by how ascending the volcano is reminiscent of changing latitude, right? A basic observation, but not many people had the opportunity to witness that previously. And we know today that globally, vegetation from deserts to rainforests are largely the product of temperature and precipitation. But as Humboldt pointed out, mountains, particularly tropical mountains, also have a strong effect on regional diversity. So here are two figures from two recent papers summarizing global richness for vertebrates and plants. And in both cases, you can see that the tropics really pop out as the richness areas, the high richness areas, but you can see very conspicuously that mountains harbor enormous richness, especially in the tropics. And that's because we think tropical mountains stack ecological niches on top of each other in a very narrow geographic space. So in this figure from a recent paper from Rabeck, you can see mountains laid out in a latitudinal gradient. On the left, low latitude, on the right, high latitude. And you can see, depicted here are the ecological niches that you traverse through as you ascend the mountain. And the problem in high latitudes is there isn't enough climatic space to support life at high elevation, such that there are fewer ecological niches. In the tropics, you can stack all of these on top of each other. As you ascend the mountain, it gets colder, climate changes, and you can fit more ecological space into a narrow geographic space. But there's also a reason why tropical mountains are different from temperate mountains, and that's seasonality, right? I live in northeastern North America, northeastern United States, and in Vermont, and we just came out of a normal winter and into a fairly normal spring. And of course, it's day and night. In fact, it snowed last week, and it's probably going to get up into the 80s next week. So plants living in my region are adapted to enormous swings in temperature. In the tropics, of course, close at low latitudes, in many, many places, the seasonal variation in temperature is very low. There's very low difference throughout the year in temperature. And that's depicted here in the two figures in this pretty famous paper by Dan Jansen from 1967, Why Mountain Passes Are Higher in the Tropics. And what he described in the paper was the difference in seasonality between tropical and temperate places, leading to the adaptation of organisms in those places ultimately has an effect on genetic isolation. So in the tropics, we expect species are less likely to get out of their ecological zone because they're adapted to their zone and it doesn't change. And therefore they're going to be more genetically isolated from each other. In temperate zones where the climate swings throughout the year, species are going to be able to tolerate that. And therefore they're probably going to be able to be more likely to get out of a particular zone and into another one, facilitating the transmission of genetic information. So with those basic ideas in mind, let's switch our focus to ferns, which is my study group. And I think they're a very good study group for the kinds of work I'm presenting here today. And here are some reasons why. They're cosmopolitan with about 11,000 species. They're most diverse in the tropics. And what I'll show you is they combine ancient history with recent diversification patterns. Now, unlike other vascular plants like flowering plants, here we don't have any distracting fruits or flowers. That's quite useful because it minimizes the plant-animal interactions to a degree. And so we think that diversity is going to more clearly reflect geographical patterns. And then also very importantly is that the taxonomy and distribution of species is relatively well known. We're not swimming in diversity such that we can't take on a large study like this. Now, many of you will think about ferns as being herbaceous terrestrial plants, and you're right for the most part. But it's important to remember that ferns also comprise one of the largest groups of epiphytes. And there's other interesting habits as well that make them a fascinating group. We have hemiepiphytes, we have scandent plants, which take over disturbances and roadsides and open areas. We have long-lived arborescent species, and there's even aquatics, floating aquatics and rooted aquatics. And then most people will think of ferns as being desiccation intolerant plants. And again, you're mostly right, but there's really a spectrum of desiccation tolerance where we also have desiccation avoiders, especially in the chylanthoid clade, and desiccation tolerant species, which are able to dry out and resurrect. And I mentioned we have an interesting mixture of diversification history. So everyone gets taught at some point that ferns are old, and they're right, but most extant species diversified after the Cretaceous terrestrial revolution. So we have become quite fond of saying that ferns diversified in the shadow of the angiosperms following this prominent 2004 paper, which demonstrated that. So a consequence of that is, on the landscape, we can find a nice mix of lineage diversity. There's some really characteristic depauperate lineages, like the horsetails or the osmundaceae, really ancient lineages with very few extant species. And conversely, we have wonderful, diverse, spectacularly diverse lineages that are not all that old. Polypodiaceae might be about 90 million years old with close to 1,200 species. These are some figures of those. Or this is a figure of species diversity in Elaphoglossum, a clade that's probably around 60 million years old with about 600 species or more. Wonderful diversity. So people have tried to depict global fern diversity at the global scale. The earliest global depiction I'm aware of is that by Hermann Christ, which he did in 1910. Christ delineated 12 floristic provinces. And what I like about his map that I'm showing you here is that he clearly saw the importance of mountains in these floristic regions. He included the Himalayan arc. You can see that there in orange. He includes, and he has it connected with East Asia. He distinguished the Andes from Amazonia. Again, it's in orange there, the Andean Cordillera. He split Brazil's Mata Atlantica in the southeast of Brazil out from the rest of South America. So he's really aware, these are all systems where mountains prominently influence the diversity, and of course he was aware of those. The next most prominent historical study that I'm aware of is that by Rolla Tryon. Here he's depicted with Alice Tryon. Rolla summarized neotropical diversity, and he did that by tabulating up monographs and floristic revisions of his time. And he produced some bubbles there on the map showing areas of richness and endemism. The bars are richness and endemism side by side. And what you'll see, what Rolla came up with is basically neotropical montane sites are centers of richness and endemism. That's what you get if you tabulate up monographs. Notice he does not include Amazonia in one of his bubbles. Amazonia doesn't appear as a fern hotspot. And then even more recently, Anna Weigand, a wonderful researcher from Michael Kessler's group in Zurich, approached global fern diversity by summarizing thousands of richness plots, transect plots done by different vegetation surveys over the years. You can see in the upper left is a figure depicting where they had plot data, and in the lower left is their map of global fern diversity, which nicely shows mountains popping out across, especially across the tropical zone. So, at this point you might say, okay, the story has been written. We know something about global fern diversity. But I think there's a lot more to be done. And that's because, in part, diversity is not just species, right? Diversity includes phylogenetic distance, divergence times, diversification rates, right? Historical processes can also be brought into this picture. So, some simple questions we might ask with a phylogenetic mindset is, well, what geographic regions are harboring rapidly diversifying lineages? Do mountains, we see that they're prominent, but do they alter phylogenetic signal in any way? Can we pick up on that? And we know that there's at least one great example of phylogenetic signal and mountain uplift story in the ferns. And that would be the Andean radiation of Jamesonia. So it's a rapid recent radiation into a habitat that is only a few million years old. And in this paper, Sanchez Baracaldo showed within about the last 3 million years, we see the diversification of the extant species of Jamesonia, which are about 45 or 60 species, into the northern Andes. And in my own work, I have found some stories that really compel me to think there's something going on or something worth investigating with elevation and diversification rates. And what we did was, in the upper panel I'm showing you, we looked at diversification rates in the Polypodiaceae. We calculated rates, and we looked at elevation at which species were occurring and how that elevational niche of a species evolves along the branches. And we found generally a positive relationship between changes in elevation and changes in diversification rate. Not a particularly strong story, but a compelling story nonetheless. And in the Etheriaceae, in the bottom panel, this is a group where we have a mostly temperate or tropical montane group, Etherium, sister to a mostly tropical mid-elevation group, Diplasium. We looked at diversification rates in that group as well. And then in the lower right panel, you can see green bars are showing you that we find Diplasium, where you'd expect it, at mid-elevations. And the green line is showing you that diversification rates, I should say richness at mid-elevations for Diplasium, and diversification rates at also at mid-elevations, peaking at mid-elevations where species richness is higher. But in Etherium, we see that richness, it peaks a little bit higher upslope, around 2,000 meters, but that diversification rates are even skewed further to the right, giving us this, interesting suggestion that, well, maybe the most upper elevation species are giving us this fastest diversification rates, suggesting that there's perhaps something going on with mountain uplift or elevation and diversification patterns in these plants. So with those ideas in mind, we set out with the following study aims. We wanted to, first we needed to paint the big picture. We wanted to characterize global patterns of fern diversity. We wanted to identify hotspots of richness and endemism like Tryon had done. And we wanted to understand factors that may be contributing to these patterns. So generally to ask how phylogeny informs these geographic patterns at the end. And I did this with two people that came out of our group here in Vermont. Jacob Suissa was an undergrad in our group. He's now working with Ned Friedman at Harvard as a PhD student, and Wes Testo was a PhD student in our lab. He's now in the Antonelli group at the University of Gothenburg. And I would say really any success of this project is owed to these two people. And here's how we approached it. We had 3 data sets. So we have which I'm showing you in the columns here. We had a set of specimen collection records, a set of ecological variables, and a set of phylogenetic relationships. This is a great time to be working on herbarium specimens because the entire world is digitizing their museums. It's spectacular. And in particular, we happen to have an NSF project to focus on ferns with 36 member groups trying to digitize 1.7 million specimens. So that is a huge help. And if you know anything about working with georeferenced specimens, you know that cleaning is the difference between real data and trash. And so the bulk of the work on this project really came down to cleaning the data and then cleaning the data, looking at what we got and realizing how to clean it better and redoing it over and over again. So we started, here's a schematic showing roughly how we did it. Cleaning data is going to be a mixture of automated and curated approaches. So this data set starts with the 1.7 million records. There is an initial automated cleaning, which gets rid of obviously problematic records. A very important step is passing through a taxonomic thesaurus. It's famously been said that a huge percentage of museum specimens are residing under the wrong name. That's true. Fortunately, it's often a nomenclatural synonym. So we can solve a lot of those problems. But really, what I'm showing you in the bottom where it says interactive maps used to flag suspicious records, that's the most critical step. We just came up with a workflow to map species distributions and look at them. And fortunately, the three of us have spent a lot of time studying the organism. So we're able to project maps and quickly say, yes, this map looks like a realistic distribution based on what we know about this organism versus that's wrong, that has mistakes, we got to check that. So our final data set had about half as many records as we started with and about 7,800 species. Now here's an example of the basic things that you're up against. So here's a clade of things that should be found in neotropical mountains or at least uplands. But I see I've got two dots in the center of Brazil, right? And these are obvious problems that you can find. One is probably rounded to the city of Manaus. One is probably rounded to the centroid of Brazil. I see one off the coast of South Africa. Somebody dropped a digit from their decimal, right? But here's the real problem is you see Australia looks like it's populated with hundreds of records that behave like a real species, right? They even follow geographic patterns. And that's a much nastier problem to watch out for where you have, in this case, it was a nomenclatural error based on a homonym causing an incorrect synonym. So that's really tricky because if you don't know this organism and what to expect, you're going to overlook these things. Well, our ecological variables come from data sets like the CHELSA dataset, soil grids, and USGS datasets. And we use things that many of you will be familiar with if you've done this kind of work, things like annual mean temperature, minimum temperature of the coldest month, soil pH. And we looked at all of these variables on their own. We also calculated a value for niche heterogeneity, which we calculated as mean within cell standard deviations of each variable where ferns occur. And then we have a set of phylogenetic relationships, and I was able to take these from our previous project where we put together a 4,000 species plastid phylogeny and then calibrated clade ages with fossils, about 22 fossils in that study. And then for this project, we recalculated our diversification rates using birth-death models in RevBayes. And fortunately, a big part of those kinds of rates, a big part of your estimation is going to be based on extant diversity. Fortunately, we have strong extant diversity estimates from a recent community classification for ferns. So nicely, a bunch of projects came together that facilitated these bigger questions. So let's look at some of the plotted results and see how they do. Here's a map of the globe, and the little black dots, they look like dust or pepper scattered onto the white countries are the individual records. And so we can start here to look at biases. Of course, we're going to have biases. In blue circles, I'm showing you places where I don't expect to see a lot of ferns, and great, we don't get a lot of ferns. That works out, checks out well. In red, I've illustrated a real sampling gap that we have, and that was India. So despite having a long history of teratological investigation and a climate that supports a lot of ferns, we aren't getting records from India because for one reason or another, those records aren't getting into the databases we have access to. So those are the kinds of problems we're up against. There's also, you can pick out, if you study this map closely, you can pick out all sorts of interesting political biases. I'm showing you in the inset on the lower right, a map from a recent paper on New Guinea, where we see a political bias, where Papua New Guinea has a pretty solid distribution of records showing the expected pattern of richness, highest in the Central Cordillera, but Western New Guinea is much less well inventoried here. We should see the Central Cordillera going right across the entire island, but it looks like it stops in Papua, which is not the case. Well, is richness biased by the number of occurrences? That's an important question. We do know that richness is going to be a combination of collection effort and digitization effort. And we know that people tend to collect a lot of stuff in rich areas. What we came up with to test how, to sort of ground truth our estimates of richness was that we came up with a proof of concept where we did a comparison of countrywide richness of our results to modern floras. So here in the blue bars are sampled richness and the orange bars are reported richness. And this worked out, this nicely gave us a sense for what we were up against. In about half of the floras we looked at, our results were within 5% of the reported diversity, which is great. I think in some cases, our result might even be more accurate. In 1 1/4 of these results, we're differing by 10 to 25%, which is a little bit problematic. Really problematic is in about 1/4 of the floras we looked at, our estimate was up to 35% different. The worst of these was the flora of China, which is particularly frustrating because it was a Missouri Botanical Garden project, and much of the data ended up in accessible databases, but some of it didn't because of the nature of that project and how data was flowing around. So that's a big challenge there. Other floristic regions that we were also had different estimates for tended to be Southeast Asia and tropical African countries. Well, looking at the results we have, we can see that richness ranges from 3 to 929 species per 100-kilometer grid cell. We recover a strong latitudinal diversity gradient, which is great. We expect that and we see it. And that's what I'm showing you in the plot on the right with records richness records peaking at the low latitudes. We also recover Africa as the odd man out. That's the old phrase people have used to refer to the fact that the neotropics and tropical Asia have higher richness. And you can see that clearly in the maps where the colored dots are depicting richness with red as the highest richness, right? Looking at elevation, we also see an interesting pattern. Now here I've binned elevation and I've binned major latitude groups. And what's interesting here is you can see across latitudes, richness peaks at mid to upper elevations. Now, but you'll also notice that it's skewed to the right in every single one. And I don't know why. I think in the tropics, that probably has a lot to do with the epiphytes, right? Ferns are have a lot of epiphytes. But why in the north and south subtropics or temperate regions, I'm not sure why I'm getting that pattern. It might be because of the patterns of precipitation, sort of the so-called horizontal precipitation, cloud precipitation that facilitate fern-friendly habitats upslope. We then took our results and we looked at weighted corrected endemism, and that's where we look at the mean of the inverse range size of the taxa represented in each grid cell. So old world endemic centers nicely match our expectations. We see the Himalayan Hengduan region pop out. We see Malaysia, including New Guinea, pop out. We see Madagascar as an endemic center. New World centers are a bit blurred together, I think perhaps because we have such high collection density throughout the neotropics. But if you squint and hunt for the red and orange dots, they do pop up in places where you'd expect them, particularly Mesoamerica, Colombia, southeastern Brazil, and Chile. And then, nicely, now we can suddenly see parallels between our results and Christ's map. I don't really know how Christ made his map other than reading, the books of his day. But, similarities pop out. The Himalayan arc pops out. The Andean Cordillera pops out. The Cape Flora pops out as a region. So this is nice to make that historical comparison. From there, we wanted to understand hotspots of richness and endemism. So we approached this by using a statistic in QGIS that works against a null distribution, where we were looked for areas of richness and endemism and areas of richness plus endemism. And when we do that, we see 8 sites of exceptional richness and endemism. And these were East Asia, the Greater Antilles, Guianas, Madagascar, Malaysia, Mesoamericana, southeastern Brazil, and the tropical Andes. But what I like about this is that now we get a sense of the uneven distribution of life on Earth, right? Which is one of our original inspirations. So what I can say now is that 79% of global species richness occurs in these 8 hotspots, which together comprise just 9% of Earth's land area. And although the figure is slightly different here, this is a similar data set. We wanted to make plots to test Tryon's hypotheses about neotropical centers of endemism and richness. And for the most part, we come up with a very similar answer. However, we also get the Greater Antilles as a hotspot. Why Tryon didn't get that, I'm not sure. Nicely, we also consistently do not see the Amazon as a hotspot, right? Okay, so that's a good start, but can we explain hotspots, right? Well, species area relationships of hotspots are real, right? We get higher species richness in bigger areas, even within hotspots. So that's a start. We expect that to be the case. But more insight comes from the idea that we can see that mountains are harboring higher richness per area than the adjacent lowlands. So in the figure here, the blue squares, the blue grid is showing you the calculated hotspot. And then the orange colored area is the land area above 1,000 meters. And when we look at richness this way, we can see within our hotspots, richness was always higher above 1,000 meters per unit area, right? And it's about 1.6 times higher in the Andes, tropical Andes. But in the Antilles, it's about 10.9 times higher. So it's really concentrated at upper elevations. We can do a similar thing for Malesia. And here, I can tell you that hotspots above 1,000 meters, what's fascinating to me is that they cover less than 2.5% of the Earth's land surface, but harbor nearly 63% of the Earth's fern diversity. So let's dig a little bit more into that. What's going on? Well, I think hotspots harbor a disproportionate amount of climate space compared to the land area occupied. So in this plot, the gray dots are all of our localities plotted with against their mean annual precipitation and mean annual temperature. And then particular hotspots have colored bubbles drawn around them for the plants from those localities. So what's amazing here is you can see the tropical Andes, right, in that teal green, comprises 52% of this available climatic space, right, for these two variables, but only 1.2% of available land area. So you're seeing just an enormous amount of climatic potential or climatic space within a small land area. And very similar to that, we see that hotspots harbor environmental heterogeneity. So environmental niche heterogeneity, I'll remind you, was calculated as the mean within cell standard deviation of each variable, okay? And in the 3 plots on the left, those are our hotspots binned out by major region of the globe, right? The colored the colored line is the hotspot, its relationship of richness to niche heterogeneity. And the gray line in each there is the adjacent non-hotspot. So within all of our regions, we find a positive relationship between species richness and summed heterogeneity. And we can also say, because we have those non-hotspots, that hotspots have more positive relationships than adjacent non-hotspots. What's going on? Probably it's just relating to the simple fact I showed you earlier that we see at low latitude tropical mountains, lots of niches stacked on top of each other. You can imagine if you're trying to sample environmental heterogeneity across the mountain in the left from 19 degrees north latitude, you're going to find a lot more niche heterogeneity than some other sites. And I think that's what's driving that pattern. I think that is also what explains why the Amazon is not a hotspot for ferns. It's flat, right? So in our data set, we recover about 2,000 species in the tropical Andes and about 400 species for Amazonia. I think both of those are a little low. I think we could find more species in both places, but it's the difference between them that matters. And I think that's probably driven in ferns because the Amazon is flat. What's amazing is, and completely different organisms, the Amazon is an enormously diverse place. And if we look at tree diversity, we're not going to see this pattern. So this is something that relates to ferns and other plants like them. And then we can ask, are diversification rates higher in hotspots? Well, we can say that mean speciation rates, so what we did was we took mean speciation rates for each grid cell based on the 3,054 species which we had in our phylogeny. So if we have a species in our phylogeny and it's plotted on the globe, we can get its diversification rate, and then we take mean rates for grid cells, right? And we're asking, is there a geographic pattern to the phylogenetic signal, or vice versa? Well, and so we might expect, are rapidly uplifting mountains full of rapidly diversifying lineages like we saw in that Paramo species I showed you at the beginning of this talk. Well, what we can say is, yes, young mountains, and we can say mountains harbor a lot of rapidly diversifying lineages, but so do other places. The pattern is not as simple as mountains harbor rapidly diversifying lineages. For example, the red arrow on the map is showing you the center of Australia. I did not expect that to be a diversification hotspot, but it's pretty hot. And probably it's being driven by the abundance of the heterosporous water ferns, Marsilia, which are they're diverse there, they're common there, they're well collected, they're represented in herbaria, they're represented with phylogenetic data. So we captured that signal there. We also might have guessed that rapidly rising mountains like the Andes would have a different signal from older mountains like those of southeastern Brazil. We don't see that pattern. There's no discernible difference when we look at it this way. We do, however, we can say that rapidly diversifying lineages have a positive relationship with environmental heterogeneity. And you might say, isn't that just because richness also has that pattern? And that might be driving a lot of it, but we also know that there are areas with low richness that have high diversification rates, like the center of Australia. So the pattern is a little complicated and bears some further investigation. So to summarize, people have been trying to depict global fern diversity for a while using different methods. We did it using herbarium specimens, and we were able to demonstrate a latitudinal diversity gradient, highest at low latitudes. We're also able to show an elevational mid to upper elevation richness curve. We were able to identify 8 hotspots of endemism and richness. We're able to show that richness is concentrated at upper elevations in hotspots, and that these hotspots harbor a disproportionate amount of climatic space, and that these patterns also have a positive relationship with niche heterogeneity. And this, I think, is probably being driven by this overall geographic pattern of niches stacked on top of each other at low latitudes in mountains. There's certainly room for other improvements. More records would be great. Better clade ages would be great. Cleaning data is so important, both taxonomy and localities. I say my personal challenge is, I think we're getting better at describing patterns, but are we closer to understanding processes? And when we can't find differences between old and young mountains, is that because ferns disperse by spores and spores are blowing in the wind and just sort of, overwhelming the signal? And this work, omits historical processes. I think next step is to combine this with analyses of ancestral areas. And last, I'll leave you with the thought that, there is a bit of urgency. Snowline has risen already 200 meters on Chimborazo since Humboldt visited there. So nothing's going to wait around and stay in stasis while we try to figure this out. Things are changing all the time. And lastly, I'd like to acknowledge a lot of people in this kind of work. I especially want to acknowledge field collectors, curators, and herbarium staff, and the people who run data portals and digitize specimens are so important for helping us put together these global perspectives based on thousands of records done by thousands of people over the years. I'm supported by the Tryon Certified Library at the Pringle Herbarium. We had help from Michael Hassler and Sean Laffin, and I thank my lab members, and I've just illustrated some of the many people who particularly have helped me over the years at taxonomic questions and collecting efforts. So I think I will stop my share because that is the end of the talk.
That was amazing. That was awesome, Michael. Thank you so much. We've got plenty of time for questions, and I will. We've got some people with raised hands. There was one question that came in early from Maria Laura Tolmos, and it asks, "Are you looking into clustering and over dispersion on the mountain areas? If so, do you expect more clustering in the tropics and temperate areas, as has been shown for trees, or should ferns behave differently?"
I love that question because it's exactly what we're going to do next. But I don't have an answer for you yet. Let's talk after we have investigated it more. I think I don't think we'll see the same patterns as trees, but I don't want to say more than that yet.
Yeah. I think, we've got a raised hand from Dr. Robert Colwell. So maybe Karen can allow him to ask his question live.
Hi, that was a terrific talk. I very much enjoyed it, and I'm a great fan of studying mountains and everything on them. But I have a question. The only time I actually saw a grid cell size was 100 kilometers square. Is that what you used for the whole study?
Yeah, great question. It is for the whole study, except the one plot where we showed the elevational richness plots. We expanded the grid cells there to help make it a more meaningful metric.
Just a second, my dog is driving me nuts. Come in, please. This is COVID time, right? Yeah. So the problem is that, I mean, if you look at Costa Rica, for example, you can fit one 100 by 100 kilometers square that goes from coast to coast and includes mountains up to 3,200 meters and lowlands and 10 kinds of habitats. And I would think the same problem would occur on other islands, sorts of places like the Antilles and the Indonesia and so on. What do you do about elevation when the square includes everything from sea level to the top of the mountains?
Well, so we can omit squares that are obviously cut off, right? But you're absolutely right. An overly large grid cell can be a problem. For example, there are areas of low richness in Central America between the Nicaraguan depression, right? Is a famous area of low richness, especially for our plants. And we really pick that up. We'll see a continuous high richness peak there. And that's exactly driven by the size of the cells. But you probably know, it's a matter of, you have challenges with large grid cells, you have challenges with smaller grid cells. So it becomes a matter of finding something that's going to allow you to do a global analysis.
Yeah. What we did in a simulation a while ago was to use a hybrid scale where more based on topography, where the more rugged the topography, the smaller the cells. So it keeps the data processing down. You can use a large cell in the Amazon, but on the slope of the Andes, you have to use a smaller one. The math is a little tricky, but it does work.
That's very cool. I will look into that. Thanks for mentioning that.
Sure. I'll send you the paper.
That'd be great.
Thanks for that question, Rob. So I'll go to another one of the written questions. Let's see. So there's one here by, I'm probably not going to get the pronunciation of the name correct, but Namju Hyo. And it asks, how would the patterns of diversity vary by genus versus species level?
That's a great question. So, we have analyzed, so is the question about diversity like richness, species, or can the question person be more specific, or are they asking about diversification rates?
I think they're asking about the diversity. Well, the question says diversity itself. So I think they're asking if the generic diversity is any different than the species level. Genera are fake, of course. We make them up. Genera are, we are the useful group name that we use to hold a group of species together. So we have large genera and we have small genera. Fortunately, this study is done at the species level, so that kind of problem doesn't
affect our results. But I may be misunderstanding your question. We do see a lot of fascinating comparisons between genera in terms of clade age, diversification rate. Again, like I said in the introduction, we have old depauperate lineages, we have young species-rich lineages. And fortunately, because ferns are mostly tropical, we capture a lot of those when we look at the tropical diversity in mountains. If I missed your question, please ask it again, and I'll try to do better.
I think we're going to see if we can get that person live to do a follow-up. So maybe in the meantime, while we're trying to set that up, there's an interesting question that's here from Sritama Badra. And it says, ferns have great variation in chromosome number and ploidy. Have you checked if the altitudinal pattern of fern species richness is correlated to their ploidy or chromosome number?
That is a wonderful question with decades of interest. And I don't know the right answer. I have seen the old evidence, was in favor of high latitudes having higher ploidy numbers, and then there was a similar report for high elevations having high ploidy numbers. Of course, a lot of the research for ferns and for many groups has a massive temperate bias. So I don't think we know the right answer for ferns, since most of the diversity is tropical and it hasn't been investigated.
So we've got a question from Matteo Radana. Is there any relation between richness or diversification rate with other non-environmental variables such as soil use or anthropogenic pressures?
Oh, yeah, it's a wonderful question. We didn't find any particularly strong signals that stuck out to us. The ones that I showed you was the best way that we could explain diversification rates. So I guess the answer is there might be something there, but if there is, we didn't really find a strong signal.
We've got a bunch of questions here. I'm going to try to get to as many of them as we can. I'm going to sneak in one of my own, though, which is you seem to explain some of the patterns of diversity in mountains having to do with the amount of environmental heterogeneity. Mm-hmm. And I wonder if there's any way of looking at how important allopatry might be in this story, and whether there's an opportunity, of course, at a smaller spatial scale, to look at, I don't know, cone-shaped mountains versus mountains with more sort of topographic heterogeneity, and whether you see differences there or not. I just wonder if you have any thoughts on that.
Well, Dov, you're, again, this is another question that has been on the forefront of my mind. And, our research group that worked on this, we have been asking these exact same questions the whole time. And since finishing this project, how can we get at that? How can we get at species interactions, species distributions, allopatry, more of a speciation question, and how can we look at the different types of mountains and how they might behave differently? We don't have an answer yet, but I would love to hear from other people who have ideas, and it's something a direction I definitely want to go in. And probably something that we want to start doing more is mapping and modeling particular species distributions and seeing how that informs our overall picture of what's going on.
Fantastic.
Yeah.
So I've got a question here from Jacob Watts. Says, hi, Michael Sundue, great talk. I was wondering what you think why you think fern diversity is much less concentrated than tree biodiversity in the Amazon. Do you think it has to do with biotic interactions or longer life cycles of trees? Do tree ferns exhibit different patterns of biodiversity than non-tree ferns?
Great question, Jacob. Nice to hear from you. I haven't studied tree diversity myself, so I've just read other people's work. My sense is that my first guess would be biotic interactions is changing the game. There are a couple other important points. Amazonia may have, in some parts has a bit of a dry season, and a little bit of drought can go a long way to lowering fern richness. So that might be also, so that there isn't necessarily one answer here. So it could be that ferns are a little bit held back. Angiosperms, with their incredible biotic interactions, have their own story going on. Tree ferns, great question. Long-lived, arborescent, quite tall sometimes. Are they different? I think there is, I'm trying to remember if the paper is published or unpublished. I think there is unpublished evidence out there that tree ferns do not follow the same elevational diversity gradient that we report here. And I think the result is, I think it's unpublished work, so I'm not going to spill the beans for those people, but I think there's a cool pattern with tree ferns, so keep your eye out for other people's work on that topic.
Well, that's fascinating. You know, this is the kind of an interesting question from Amjad-ur-Rahman, and it says, is there anyone interested in exploring the turtophytes in Western Himalayas in Pakistan? You know, it sounds almost like an offer. I don't know, but
yeah, email me. Let's talk. If you're interested, let's see what we can do. I don't know a lot of people from that particular region, in particular. I would love to make new friends.
You know, let's go live to Hannah Tumisto, who's got a question. We'll let she's raised her hand. She obviously knows something about ferns.
Yes, she does. I can't wait.
Okay. Hello.
Hi.
That was really great talk, Michael. I enjoyed that a lot.
Thank you.
And I couldn't kind of resist putting my hand up when he started talking about the differences between Amazonia and the mountains.
Yes.
And I think you did hit one of the important things there with that dry season thing, because I think I have the impression that the highest diversity of ferns is in the mountains, in those zones that have pretty much a continuous cloud cover. Mm-hmm. So they're kind of very, very moist forests, and that's where you get these ridiculous amounts of the epiphytes. And especially that diversity is much lower in Amazonia. And we do tend to find that when you get the dry season, it takes the diversity down a lot. And then the other thing that might have an impact is the soil conditions, because much of Amazonia has pretty poor soils. And that's another trend that we have found, that in poorer soils, we tend to get fewer ferns per unit area. And also in total, the species pool on poor soils tends to be lower. And I think these, especially things like Andes, which is a recent mountain chain and has kind of marine deposits in its core and volcanic stuff. And so it probably has pretty fertile soils, which probably means that it's pretty good substrate for ferns to grow on. That's at least the kind of impression that we get from Amazonia. So that might be another reason why the Andes is so species rich.
Right. I was thinking of your work in the back of my mind as I was answering some of these questions. So, we didn't find soil patterns that were so strong that we, emphasize them. But I think that has to do with the scale at which we're working, right? And in your work, you guys have documented wonderful patterns of the influence of soil chemistry on fern distributions. And I think tell me if you agree, I think it's probably because you're working at a different scale. Those studies were within Amazonia, strictly on Amazonian ferns. So you're much more likely to see these differences that at the big picture would be very subtle.
Yeah, I think it's definitely got to do with the length of the environmental gradient that we're looking at. Because within Amazonia, we're kind of within a relatively uniform climate area, even though we have differences in seasonality. Mm-hmm. But in the Andes, just like you were showing recently, there's a huge variation in climate in very short times. And then you get these complicated cross variations between different kinds of climate conditions, different kinds of seasonalities, and then you get the very complicated geology. Mm-hmm. In addition. So then it's probably the climate pattern that is easiest to see. And then the kind of soil influence gets kind of drowned. Whereas in the Amazonia, it's the other way around, because the climate is uniform over very large areas. Then what we tend to see more easily is the soil influence.
Mm-hmm.
Now that's fascinating. I mean, where you have the power to see what sort of factors that might be important could be really important at these spatial scales, which is super cool. So we're almost out of time. I'm going to go to one last question. Hannah, thank you very much for speaking up and having that dialogue. This last question is from Gabriella Zukim, and I think it's an important one. How do you deal with potential taxonomic misidentifications in GBIF data or other data sources? that's the question. And I think it's pretty fundamental to these sorts of endeavors. So curious to see what you think.
I love that question. Thank you for asking it, Gabriella. I remember we met a few years ago, and you are exactly the right kind of person to ask that, because I know you work hard on these questions and you care, and I'm with you. It is, you know, if you don't have good taxonomy, none of this works. If you don't have good species concepts, everything goes wrong quickly. I think when I started working at this scale, I was afraid that that kind of challenge would tank the whole project. What I have come to discover is actually I feel like herbaria of the world are largely populated with well-studied and carefully identified plants, much more than I would have thought. We went through a lot of effort to find obviously wrong things, right? By combing through maps and distributions. That said, it's easy to overlook a lot of misidentified things, and that's going to influence the work. And anyone working at this scale is going to have that kind of problem with their data set. And when you have very cryptic or similar looking species that have the same distribution, you're not going to be able to find those combing through GBIF records. So I think it's an ongoing challenge. I think if it was I don't think it's problematic in this case to the degree where we received results that were meaningless, right? We had expectations of richness and diversity patterns, and we recovered those. So we didn't find, a lot of patterns that were really astonishing or weird or unheard of, which you might have gotten if, for example, every plant from Hawaii was misidentified or something. Hawaii would blow up in your results as some weird aberration or disappear or something. We didn't see those kinds of effects, at least in the published data set. In the raw data we did, for example, hundreds of records are mapped to what I can either imagine is either a botanical garden in Sacramento, or maybe it was the fern grower Barbara Jo Hoshizaki's personal garden. That one locality was in the raw data had enormous species richness of all sorts of things that of course definitely are not native to that area. And if we hadn't found those kinds of problems, we'd be overlooking it. But that's a geographical location curation question. You're actually asking about species taxonomy, which is a trickier problem. And I can't say more than what I have just walked us through here. I think it's ongoing, it's important, but I don't think it inhibits the entire process.
So Michael, I just want to thank you from the International Biogeography Society for giving this fascinating talk and taking the time to answer the questions. It was just fantastic. And I want to thank everyone for tuning in. We can't all applaud, but thank you so much. And I hope everyone has a wonderful afternoon or evening, and tune in about a month for our next talk by Jessica Blois. Should be fascinating. All right, thank you. Take care, everyone.
Thanks, everyone. Bye.